Hi everyone, today we'll be solving chapter 22, DNA biology and technology, from the book Human Biology, the 14th edition, belonging to the author Sylvia S.Mader. Question number 1. The enzyme responsible for adding new nucleotides to a growing DNA chain during DNA replication is DNA polymerase, kinda like the construction worker. It is the one that adds the nucleotides to the growing strand. Question number 2. This process is responsible for the formation of a new DNA helix. DNA replication, because it's DNA replication, it's a semi-conservative, in which what is semi-conservative is that basically the new DNA, it will have one of the old strand of DNA and a new strand. Transcription and translation are processes involved in DNA protein synthesis. Okay. Coming to question number 3. RNA is different from DNA in which of the following ways? RNA is generally single-stranded. Yes, that is correct. Although sometimes DNA can also be single-stranded, like in the case of viruses. That's why the word generally. Option B, RNA contains uracil rather than thymine. This is true. The sugar of RNA is ribose. True also, while as in DNA you have deoxyribose, in which the second carbon on the deoxyribose doesn't have a hydroxyl group but rather a hydrogen. RNA, option D, RNA is translated to form proteins. Yes, this is true. So all of these are correct. Question number 4, RNA processing. Okay, in other books, it can also be called RNA splicing. So what it does is that it removes the interns and leaves the exons. So it removes the exons, leaving only the interns. Opposite actually happens. Is the same as transcription? No. Transcription is when an mRNA is formed from the template strand in the DNA. Is an event that occurs after RNA is transcribed. Actually, it occurs after RNA. Yes, after it's transcribed and before it is translated. So immediately after the formation of the mRNA, it will immediately after transcription, it will basically remove the interns and it will leave the exons, and then it will travel outside and via the cytoplasm into the ribosome. Is the rejection of old worn out RNA? No. So this is option C is correct. During gene expression, an anticodon of a tRNA pairs with. So anticodon is basically like we have three types of RNA. We have mRNA and tRNA. And it is basically like the one that will go and bring the amino acid, the correct amino acid according to your mRNA that you have. So tRNA, it pairs with amino acids in the polypeptide. The top part of the tRNA, it's attached to an amino acid, but the anticodon, which is like the bottom part, which will form hydrogen bonds with the complementary bases of the mRNA. I think I already gave an answer, so it will be DNA nucleotide bases. No. RNA nucleotide bases. No. mRNA nucleotide bases. For example, I have in the mRNA I have UCG. The anticodon for it will be AGC. And AGC, for example, I do not have a key with me right now, but for example, it indicates a certain amino acid. For example, just let's say it's valine, for example, or alanine, or glycine. Just let's say glycine. So here you will have glycine. This is just a visual representation of what's happening. Question number 6. Following is a segment of DNA molecule. Remember that only one strand is transcribed. Template strand, non-template strand. What are the mRNA codons? Okay, so template strand is the one that we will focus on. So as you can see, it is TGA. So obviously it will be mRNA, it will be with T, it will bear with A. GC, AU, remember, we don't have thymine in an mRNA. GGA, it will be, I'm complete writing it here, it will be C, it's going to be C, C, and this time it's going to be U, and so on. As always, we know that C is with G and A is with U in the case of mRNA. Now, the tRNA anticodons. So, for example, this one is CCU. The tRNA, it has thymine, so it's going to be GGA. And then option codes, part C of the question is asking for the sequence of amino acids. So you'll have a key and you will see GGA stands for which amino acid, and then it will be your amino acid. Question number 7. Which of the following processes occur in the nucleus and forms a complementary copy of one strand of the DNA molecule for gene expression? So I want a complementary copy of the strand of DNA molecule for gene expression, and this process occurs in the nucleus, so it is transcription. A little trick my biology teacher taught me is that to not confuse yourself about translation and transcription, just look at the L and the C. Which one comes first alphabetically? The C, of course, so transcription occurs first and then translation. Question number 8. Restriction enzymes found in bacterial cell are ordinarily used to during DNA replication to degrade the bacterial cell DNA, to degrade viral DNA that enters the cell, to attach pieces of DNA together. So actually in nature, restriction enzymes are found within bacteria, and they're used as a defense mechanisms to viruses to basically cut their DNA. Question number 9. Which of the following is a benefit to having insulin produced by biotechnology? So prior, in the past, the insulin used to be produced or extracted from pigs, mainly from animals, and some people just had allergies. A lot of animals were slaughtered, and the amount wasn't that much. The demand was still higher than the supply. So now we produce insulin by biotechnology, and one of the benefits, list of the benefits, is that it's just as effective. And right now we like put in bacteria in a small like container, you will get a large amount of insulin, so it can be mass produced, and people don't experience allergic reactions, and it is indeed less expensive than slaughtering animals. So all of these are correct. Question number 10. In this process, a gene of interest is inserted into the chromosome of a bacterium, allowing the gene to be expressed within the cell. So this is basically the process of genetically engineered bacteria. Let's take the example of insulin. I will take the gene of insulin, for example, from a human cell. I will take the gene for insulin and I will add this gene into the bacterial plasmid. I will also add a transcription factor. What is a transcription factor? It's just basically something that will like stimulate the bacteria to produce this gene, and so the bacteria, it will carry out the process of transcription translation, and just it will express this gene, so insulin will be produced. This process, it's called allowing the gene of interest inserted into chromosomes, allowing the gene to be expressed within the cell. BCR, no. DNA cloning, no. DNA replication, no. DNA sequencing, microarray technology. So all of the options, they're not what I have for the term. I normally got used to the term being just genetic engineering. So let's look at words closer to that. Microarray technology, it's a whole another process. DNA sequencing, you're basically looking at the base, the nucleotide bases in the DNA. DNA replication, no. PCR, it's a process when you take a small piece of DNA and just basically amplify it. DNA cloning, it's one that is most suitable. And other, it's just basically like recombinant DNA. Question number 11. In this process, cells are removed from the body and a vector is used to insert a gene of interest. So I'm removing cells and then I'm using a vector to insert a gene of interest. Fingerprinting, no. Ex vivo gene therapy. Ex vivo gene therapy. It can be in vivo gene therapy. DNA sequences. DNA sequencing. I will go with ex vivo, just it makes sense. X is always out something external. Question number 12. This field of study examines the function and interaction of proteins within a cell or an organism. So proteins, not genes, genomics, bioengineering. This one. Question number C. This was it, and thanks for watching. If you have any question, please write it in the comment.